Okay, so how we can trigger an immune response to react against the cancer. So basically, on the surface of cancer cells, there is proteins that are termed as tumor-associated antigens, or we denote it as TAA. This TAA is highly populated on cancer cells compared to normal cells, and this TAA can induce specific immune response. So basically, it is possible to develop a therapeutic cancer peptide vaccines using TAA as the vaccine antigen, in which this TAA can be recognized by cytotoxic T lymphocyte or CTL. So that's when we deliver cancer vaccines that containing this TAA as an antigen, our immune response will react to it, and once it recognizes and can process this TAA, and our immune cells, mainly the CTL, will start to react on cancer cells that already have the TAA on its surface. And consequently, it will lead to the killing of the cancer cell. So basically, this is the theory of how the therapeutic cancer vaccines can work. So this is just a further explanation on what I have mentioned just now, in which the CTL can recognize the tumor antigen as a marker which only presents on the cancer cells but not on normal cells, and this can lead to our immune cells to recognize the cancer cells and can induce the activation of cytotoxic T lymphocyte immune response and subsequently kill this cancer cell. So CTL is the immune response that is triggered by the activation of T helper 1 cell. As you can see here, the CTL is part of the cellular response. So why we need a cellular response compared to humoral response is because that the cancer is an intracellular based disease. So we need a cellular immune response in order to be effective. It needs to be triggered in order to be effective to induce the immune response against cancer cells. So if you can see, this is the pathway on how we can activate the immune response by the help of adjuvants. And these adjuvants can also be the viral vector that can be used to deliver specific antigen, and in this case is the cancer antigen based on the TAA, which is the tumor associated antigens. So this is another approach on how cancer immunotherapy can be formulated based on viral vector. So firstly, we can remove the normal white blood cells from the cancer patients, and then we can infect these white blood cells with retrovirus that already contain the gene of interest, in which these genes enable the white blood cells to make T cell receptor that can stick to cancer cells. And after the white blood cells are reintroduced, we're putting it back into the patients, the T cells bind with the cancer cells and further destroy them. So this is more like a personalized immunotherapy for cancer, which you use the similar patient and we put back something that we modified or engineered to kill back the cancer cells. And another approach that using virals is, we can call it an oncolytic virotherapy, which is we utilizing the use of replication competent virus to destroy cancer, which means that the virus has the ability to replicate. But the viruses are designed to be capable of selectively replicating in tumor cells, which lead to their destructions, not on normal cells. So the simple concept is that we deliver something, which is the virus, which have the ability to only replicate in cancer cells. And once it replicates to cancer cells, it will produce more and more oncolytic virus, and this oncolytic virus will target more and more cancer cells. Once it replicates, it will destroy it and destroy the next cancer cells. So this means that this treatment is that we design the virus to act as a drug that can kill the cancer cells on its own. So this is just the diagram to see on how the oncolytic virus can be designed to only replicate in cancer cells. So the virus selectively replicates in cancer cells because these cells sacrifice a great deal of their immune function in order to avoid cell death. So that's all from me, and thank you and good luck for your exam. Thank you very much.